The previous E. coli gene regulatory network example was mostly a static example, although by way of the perturbation analysis, one can think of artificially evolving the network one step at a time to see how the network can evolve over time or as a result of it interacting or being perturbed by some other system from the outside. But we can move to a more dynamic example. In this case, the network associated to a type of cell that is called T-naive cell, which is associated to the immune system, and is a cell that has not yet been fully differentiated. In this new regulatory network, we started from a network representing this undifferentiated state and followed it to its differentiated state when it becomes a differentiated Th17 cell, which is a specific cell of the adaptive immune system that helps keeping the organism from getting, for example, an infection, and is related to all sorts of interesting immune processes. So this cell was analyzed in detail in a paper published in Nature in 2013, and the data was made available from that source. What we did was to do exactly what we did for E. coli, but now on 3 networks representing the cell as a dynamical system evolving from a non-differentiated to a differentiated state. You can see on figure A how the algorithmic information of genes distribute and how many of the genes are negative and positive, meaning that they can move the cell towards or away from randomness. When we talk about the cell, we are talking about its genetic network representation, which is a good representation of the cell as it determines many, if not most, of its most basic processes, leading to protein production and metabolic pathways. But of course, it may not represent every aspect of the cell. So on figure A, you can see how genes distribute differently at every step, with the 2 first cases closer to each other than its final configuration. Now, as you may remember, we have been saying that systems that have negative and also positive elements are more reprogrammable because, as we found out, they allow us to move the dynamical system qualitatively, namely to modify the attractor landscape by increasing or decreasing the attractors, and therefore the depth of such attractors. One can see how in the differentiated state, colored in green, the cell distributes all its genes on the positive side, meaning that it has reached some sort of both dynamical but also algorithmic stability in line with all we saw in the previous unit. Moreover, if we were doing any of this by chance, we would not find any overrepresentation of the genes that we marked as negative or positive when performing a gene ontology enrichment analysis. However, we did find that genes related to processes identified in this kind of immune cells were pinpointed by the algorithmic dynamics analysis, as you can see on figure B. So in figure B, we have 3 plots corresponding to each differentiating time, indicating how many genes identified as the most positive or most negative appear in the literature as associated to the process of differentiation. If we had not captured the right genes, the distributions would look flat, as if we had chosen random genes out of all possible genes in the network. But genes that are more positive or more negative appear with greater number in the early stage, and in the intermediate stage, genes in the positive patch were highly informative of differentiating processes. And finally, in the last stage, again, the distribution is far from uniform, indicating that those genes identified by algorithmic dynamics were involved in the processes of the differentiation of the cell. In the next slide, we will explain how this gene enrichment analysis was done in some detail. But in figure C, you can see the distribution of genes that were positive and negative and how they flipped from stage 1 and 2 and then became almost all positive in the last stage, colored in blue, almost like locking the state of the cell when it reached its differentiated state. Finally, on figure D, we can track the overall complexity of the network, in which we see, also as suggested by algorithmic dynamics, that the first network somehow received signals to start its differentiating process, probably captured in its increase of complexity in the second stage, where the estimation of algorithmic complexity increases according to BDM, and then decreases again once differentiated. Here is a Venn diagram in the form of a network showing the genes in square nodes that have been identified in the literature from 9 major papers shown in black elliptic nodes covering research on Th17 cells. We had to do this because traditional gene ontology databases are not specialized enough to cover literature specific to an immune cell like this one, even that this is at the forefront of current research. For the creation of this network and choosing the literature on which it is based, we teamed up with professional immunologists. These papers cover the majority of genes that have been associated to Th17 cells. Linked genes in the figure are genes found in common between 2 or more papers. Black lines show the number of genes found in common between every 2 papers, with the thickness denoting the size of the overlap that were used in the gene enrichment analysis of the previous Th17 differentiation network. But even more interesting, to see if we are doing what we are claiming to do, we devised a third experiment after getting the expected results from a highly curated and validated gene regulatory network from E. coli and then the Th17 network. This other experiment involved taking the gene regulatory network of 21 cell types, mostly based on experimental results and therefore of high quality. We then applied not only the algorithmic information dynamics but also the reprogrammability indexes, hoping that we would be able to tell in what stage of differentiation every of these different cell types were. The experiment makes a lot of sense if you think about it, because all cells ultimately come from the same very first cell that we call an embryonic stem cell, denoted by the letters ESC, from where all other cells come from. And what we found was not only that the embryonic stem cell was on top of the programmability measure, as you can see on both diagrams, but also that another type of stem cell called hematopoietic stem cell, here denoted by the letters HSPC, there are stem cells that give rise to all other blood cells, had also a high reprogrammability as it would be expected, and that for example B and T cells followed closely and were relatively close to each other, conforming to the biological expectation, and then all other differentiated cells were on the other side of the reprogrammability index with low algorithmic complexity. So by taking this information, one can attempt to reconstruct an attractor landscape, also commonly known as Waddington's energy landscape, where, similar to what we saw in the unit of dynamical systems, one can capture and picture the attractor landscape of an embryonic stem cell and place the non-differentiated state as being on top of a hill in an unstable position where moves can make it differentiate into different cell types by falling into different attractors represented by valleys that are also more stable than the hills. So I hope you see how interesting is the application of algorithmic information dynamics to the real world, in particular to molecular biology and genetics. In the next lectures, we will see other applications to other areas such as evolution, cognition, and machine learning, among other topics.